second cytoskeleton in today's session is microtubules. First, we will focus on the structure of microtubules. Microtubules are one of the three principal components of cytoskeleton. A microtubule is composed of tubulin dimers consisting of two closely related polypeptides called as alpha tubulin and the beta tubulin. The tubulin dimers, this much portion if you consider, one blue and one green together is a dimer. Okay, this way if I draw, this will be your tubulin dimer. It consists of a green colored alpha tubulin and a blue colored beta tubulin. So tubulin dimers polymerize into protofilaments. This protofilament is assembled around a hollow core. So we have a hollow core and above on which the protofilaments are assembled. For the different monomeric subunits, that is the alpha tubulin and beta tubulin, are arranged together to form a polymer. This polymer is called as microtubule. The alpha beta heterodimer arrange themselves in head to tail manner, forming a protofilament. 13 such protofilaments combine to form microtubules. So someone can ask you a very important MCQ, a simple one, but to get the facts right, how many of protofilaments are required, or how many protofilaments combine to form a microtubule? Your answer invariably should be 13. When you talk about the structure of tubulin dimer, we have to understand based on this diagram that each tubulin subunit has a binding site for GTP. Now let's look at the diagram of alpha tubulin. The alpha tubulin will bind with GTP irreversibly, that is GTP cannot be hydrolyzed into GDP. Why? Why it cannot be hydrolyzed? Because the GDP is bound, or it is trapped in the interface between alpha tubulin monomers. So we have alpha tubulin monomers and this GTP is trapped initially between the alpha so this is trapped between the interface between alpha beta tubulin monomers. So alpha tubulin binds with GTP reversibly, that is GTP cannot be hydrolyzed into GDP. As the GTP bound is trapped, as I have marked with a red pen, it is trapped at the interface between alpha beta tubulin monomers. Let's focus on the role of GTP in microtubule polymerization. Before I go to the figure, before I go to the explanation mode, let's understand the figure. If you see the microtubule also has a plus end and a minus end. On the plus end, we have GTP bound microtubular tubulin dimers, and on the minus end we have GDP bound. So let's see. Tubulin dimers with GTP bound to beta tubulin associate with the growing end or the plus end. Where they are flat sheet, you can see this is a flat sheet. which zips up into the mature microtubule, which will increase in height into a mature microtubule just behind the region of growth. So based on this, we have shortly after polymerization, the GTP bound to beta tubulin is hydrolyzed to GDP, and since GDP bound tubulin is less stable in the microtubule than the dimers at the minus end, they rapidly dissociate. So again, just like actin, only thing is that actin had ATP on the plus end or the barbed end, your tubulin has GTP at the plus end. So considerable amount of association of GTP bound tubulin dimers take place, but as we move from plus to minus end, this GTP removes phosphate and gets itself converted to GDP. Since GDP bound tubulin is less stable in the microtubule at the dimers at the minus end rapidly dissociate. Since dynamic instability of microtubules, very important concept. Dynamic instability results from the hydrolysis of GTP bound beta tubulin during or shortly after polymerization, which reduces its binding affinity for adjacent molecules. So what I try to explain over here, or what the figure wants to imply, is that plus end always the tubulin dimers would join with GTP, but as we move from plus to minus end, the GTP gets hydrolyzed by releasing a phosphate, and after hydrolysis the GTP bound microtubule dimers are unable to maintain the polymerization. So we get a situation where GTP bound microtubule leads to association, and that too from the plus end, and GDP bound microtubule undergoes dissociation from the minus end. Okay. So this is about the dynamic instability of microtubules. Further, the growth of microtubule continues as long as new GTP bound tubulin molecules are added more rapidly than GTP is hydrolyzed. So if the number of GTP bound, if the number of GTP bound dimers, the addition is more than that of the dissociation, only then the GTP cap is retained at the growing end and the hydrolysis takes place. However, if GTP is hydrolyzed more rapidly than new subunits are added, the presence of GDP bound tubulin at the plus ends of the microtubule leads to disassembly or shrinkage. So what is disassembly or shrinkage? If GTP is hydrolyzed more rapidly than new subunits are added, even before the next subunit is added, it becomes hydrolyzed. In that case, what would happen? That the plus end of the microtubule leads to disassembly and shrinkage. What is the role of microtubule associated proteins in dynamic instability, that is growth? First, the dynamic behavior of microtubule is regulated by microtubule associated proteins, also called as MAPs. These MAPs will bind to their plus ends. And we have some of the proteins, say for example polymerases. What the polymerase does, the polymerase will accelerate the growth by increasing incorporation of GTP bound tubulin. On the other hand, we also have a phenomenon called as catastrophe. In contrast, if it is not polymerase, if it is depolymerases will dissociate GTP bound tubulin from the plus end, leading to microtubules shrinkage, and this microtubule shrinkage is called as catastrophe. The microtubule shrinkage is called as catastrophe. Also, we have another protein, the class proteins. The class proteins will rescue microtubules from catastrophe by stopping disassembly and restarting growth. Let's understand the intracellular organization of microtubules as a part of the cytoskeleton talk that I am delivering. At the very outset, we can see two different types of cell, one at interphase where the cell is not dividing, and another is a mitotic cell wherein the spindle fiber formation is very clearly seen. If you see the interphase cell, in animal cells, most microtubules extend outward from the centrosome. This is the centrosome, and you can see all the plus ends of the microtubule extending out of this centrosome. In a non-dividing cell such as the interphase cell, the centrosome is located adjacent to the nucleus. Very clearly, I will just shade the nucleus. This is the nucleus. Since the cells are not dividing, just adjacent to the nucleus you have, this is the nucleus, adjacent to the nucleus you have the centrosome. Now we see what happens during mitosis. During mitosis, the microtubules similarly extend outward from the duplicated chromosomes. Here the chromosomes have duplicated and these project outward. The microtubules project outward from the duplicated chromosomes to form mitotic spindle. This mitotic spindle is responsible for the separation and distribution of chromosomes to daughter cells. So these are the spindles which break down and forms two different entities. That is possible only by the mitotic spindle, which is responsible for the separation and distribution of chromosomes to daughter cells. Let's recapitulate. We have the centrosome towards the center in undivided or interphase cell. Now the spindle fiber has stretched. The mitotic spindle has brought the separation of the chromosomes to individual daughter cells. The centrosome thus plays a very key role. The centrosome's role is as a microtubule organizing center. The microtubule organizer center, and this organizer center helps to determine the intracellular distribution of microtubules in animal cells. Plant cells do not have organized centrosome or other form of microtubule organizing center. Instead, the microtubules in most plant cells are dispersed and form an array of underlying plasma membrane. Cytosome also serves as an initiation site for the assembly of microtubules in animal cells, which then grow outward towards the periphery of the cell with the minus ends still anchored in the centrosome. You don't see minus. Why you don't see minus? Because most of the minus overlaps with the area where the centriole is or centrosome is located. We now come to the effect of colcimid. In one of the experiment, the colcimid was given to animals. What colcimid does? Colcimid disassembles the microtubules. The cells was treated with colcimid for 1 hour to disassemble the microtubule. The drug was then removed and the new cell allowed to recover for 30 minutes, allowing the visualization of new microtubules growing out of the centrosome. The drug was then removed, so we can always plan in advance to go for the hunt for the drug or the hunt for pathway which will definitely lead us to success. Therefore, the new microtubules growing out of the centrosome, these days it is very common, it is seen, but then it was a big challenge. Again, intracellular organization of microtubules. The minus ends of the microtubules are anchored in the centrosome. Once again, let me brief you with the pen. This is the centrosome. You don't find any minus. Why you don't find any minus? Because these minus are all embedded within this centrosome region, so you don't find any minus. In the interface cells, that is, are the interface cells. The centrosome is located near the nucleus. There it is. The centrosome, already highlighted in red, is near the nucleus, and microtubules extend outward, and you can see microtubules originating from minus end of the centrosome but extending outward and located near the nucleus, and the microtubules extend outward to the cell periphery. During mitosis, the duplicated chromosomes separate. There it is separating. During mitosis, the duplicating chromosomes separate and microtubules reorganize. The microtubules will reorganize to form mitotic spindle. What are some of the key proteins in centrosome? The key protein in centrosome is gamma tubulin. Again, students, gamma tubulin is a species of tubulin. This gamma tubulin serves to nucleate the assembly of microtubules, that is stitched together. Gamma tubulin is associated with 8 or more other proteins in a ring shaped structure called gamma tubulin ring network. So, it can be associated with 8 or more other proteins in a ring shaped structure called gamma tubulin ring complex, and this gamma tubulin ring complex acts as a seed for rapid microtubule growth. Some additional points with respect to the centrioles and centrosomes. Centrioles are necessary to form cilia and flagella. We will talk about it in the subsequent part of my talk. They are also not needed for microtubule organizing functions of the centrosome and are not found in unicellular eukaryotes and most meiotic animal cells such as the mouse eggs. It is pericentriolar material, not in centrioles, that contain gamma tubulin ring complex and functions to initiate microtubule assembly with anchor microtubule minus ends. This segment, that is organization of microtubules in nerve cells, provides very good example of the association of stable microtubules with cell polarity that is provided by nerve cells, which consist of two distinct processes, one being axon, another is dendrite. Just to recap, axons are processes that carry impulses away from the cell body, that is from cell body away, whereas dendrites carry impulses towards the cell body. Now both axons and dendrites are supported by stable microtubules together with the neurofilaments that shall be discussed in the last segment of my talk, because first segment was microfilaments, especially actin. Second segment that I am talking right now is microtubules, and in the third segment I will be talking about intermediary filaments. Stable microtubules in both axons and dendrites terminate in the cytoplasm rather than being anchored in the centrosome. We all know that in most animal cells the plus end was in the centrosome, hidden behind the centrosome, and the minus end projected outside. But here the stable microtubules in axons and dendrites, they both terminate in the cytoplasm rather than being anchored in the centrosome. Focus on the area that I have highlighted or put a red box. In dendrites, the microtubules are oriented in both directions with their plus ends pointing both towards and away from the cell body. So both towards and away from the cell body, the microtubules are arranged. In sharp contrast, in axons, the microtubules are oriented with their plus end pointing towards the tip of the axon. So there you have the plus end, here you have the minus end. Microtubule associated proteins cap both plus and minus ends and then stabilize the microtubule by binding along their length. Now there is a difference in protein between the two processes. In fact, we have MAP2 in dendrites, whereas tau in case of axons. So in dendrites we have MAP2, right now I am highlighting, and tau in axons. Tau in axons, that's tau in axons. Well, in this segment we will be focusing on the microtubule motor proteins. Those are dynein and kinesin 1. We have already come across a motor protein, especially myosin 2, as a representative example of the microfilament or actin. Okay. Now we come to two motor proteins which comes under the category of microtubule. One of course is dynein, another is kinesin 1. Both dynein and kinesin 1 move in opposite directions along the microtubules, that is towards the minus and plus ends respectively. So these are two types of microtubule motor proteins, and the first of these microtubule motor proteins was identified as dynein, which was isolated by Ian Gibbons in 1965. The purification of this form of dynein, called axonymal dynein, was facilitated because it is a highly abundant protein in cilia, just as abundance. So what's exactly the structure? First we will go with dynein. Dynein consists of 2 or 3 heavy chains. Of here of course 2 are shown. So 2 heavy chains, these are the 2 heavy chains, and multiple light and intermediate chains, multiple light, these are the light and intermediate chains. There are multiple light and intermediate chains that you find. The globular head domains of heavy chains are motor domains. So these are the heavy chains, you have the globular head, and those globular heads are the motor domains of dynein, which also leads to movement. Coming to kinesin 1, kinesin 1 consists of 2 heavy chains wound around each other in a coil structure, and the 2 heavy chains are shown in orange and chrome yellow. The globular heavy domains of domains of heavy chains bind to microtubules and are the motor domains of the molecule. So the head domains are also the motor domains. Transport of vesicles along microtubules. Kinesin 1 and other plus end directed kinesins transport vesicles and organelles in the direction of microtubule plus ends. I will highlight these plus ends, the microtubule plus ends, everyone learns to do to the plus ends. So kinesin 1 and other plus end directed kinesins transport vesicle and organelles in the direction of microtubule plus ends, which extend towards the cell periphery. In contrast, dynein and minus end directed members of the kinesin family carry their cargo in the direction of the microtubule, that is minus ends, which are anchored in the center of the cell. With animation, I think things will be still better. Let's understand how.
Kinesin is a motor protein that moves vesicles or organ.
Kinesin is a motor protein that moves vesicles or organelles towards the plus end of the microtubules. Kinesin contains 2 heavy chains that are wound around each other in a coil-like structure and 2 light chains. The globular head domains of the heavy chain bind with microtubules and are the motor domains of the molecule. We now look into the structure of cilia and flagella. As you can see, inside the cilia and flagella is a microtubule based cytoskeleton called as the axoneme. The axoneme of a primary cilium typically has a ring of 9 outer microtubule doublets, also called as 9+0 axoneme. So we have 9 doublets of axoneme. When it comes to the axoneme of a motile cilium, in addition to the 9 outer doublets, you also have 2 central microtubule singlets, and together this arrangement is called as 9+2 axoneme. Here I have, for the benefit of students, I have even given a count: 1, 2, 3, 4, 5, 6, 7, 8 and 9. So 9+2 axoneme. This axoneme acts as a scaffold for axonomal inner and outer dynein arms that move the motile cilia and provides track of molecular motor proteins such as kinesin 2 that carry protein along the length of the cilium. Now let's see some of the features. The central tubule is enclosed by projections. You have central sheath. This central sheath is connected to A tubules of peripheral doublets by radial spokes. Dynein bind with A tubule. The doublets are connected to each other by interdoublet bridge. Interdoublet bridge is an elastic protein. The best example is kinesin. The radial spokes in groups of 3. Finally, you have radial spokes in groups of 3. Now, these are the structures of primary and motile cilia. You have the primary cilium, as we discussed earlier, doesn't have only 9, not 9 plus 2, whereas motile cilium has 9 plus 2. The tensile strength is much more in case of the motile cilia. To end the talk, at least to end this segment, we have discussion on some of the drugs that affect microtubules. First is Taxol. Taxol is anti-cancer drug. It stabilizes microtubules and prevents polymerization, depolymerization. Colchicine binds to tubulin and blocks polymerization. Microtubules depolymerize at high colchicine concentration. Vinblastine and vinblastine, these are the one which binds to beta tubulin and prevent polymerization. Then you have nocodazole. Nocodazole causes depolymerization of microtubules by binding to beta tubulin. Since we just completed microtubules, now it's time for some questions based on microtubules. Here goes the first MCQ. The microtubule binding activity of MAPs, that is microtubule associated proteins, is controlled by option A phosphorylation, option B oxidation, option C reduction, and option D hydrolysis. Of course, the microtubule binding activity of MAPs is controlled by phosphorylation and dephosphorylation. The correct option is option A. How many protofilaments are present in single mammalian microtubule? We already saw 9 plus 2 arrangement and we have 13 protofilaments. So, the correct answer is 1, 3, 13 protofilaments. This is a question from CSIR Net December 2018. A single protofilament of a microtubule grows at a speed of 2 micron per minute. Considering that there is no catastrophe in the microtubule nucleation and the size of the tubulin unit is of the order 5 nanometer, how many tubulin units are added to the growing microtubule per minute? 4 options are there: 400, 1600, 3200, 5200. The calculation first. Each microtubule is made up of 13 protofilaments. Now the size of the tubulin is 5 nanometer, which is given. What is the rate of the speed or rate of flow is 2 micrometer per minute, which comes to 2000 nanometer per minute. Now number of tubulin units added per minute is 2000 divided by 5, that comes to 400. Now 13 protofilaments are there, so you multiply 13 by 400 into 400, you get 5200, which is a fantastic question that has been framed. The major microtubule organizing center in most animal cell is kinetochore, nucleus, kinetochore, nucleus, centrosome, centromere. The correct answer is centrosome. Which microtubule associated motor protein is responsible for movement of cilia? Kinesin, niacin, myosin, dynein. The correct answer is dynein. In motile cilium or flagellum, the microtubules are arranged dash. There are two blanks you have to fill, 13 in a circle, 9 triplet in a circle, 9 doublet in a circle around a pair of microtubules, 2 microtubules perpendicular to one another. The correct answer is 9 doublet in circle around a pair of microtubules. Dynein and kinesin, which of the following statement holds good? Dynein and kinesin are two subunits of microtubules. They are motor proteins that generate movements associated with microtubules. The motor proteins that generate sliding of the sarcomere create the crawling motion typical of amoeba. What is it? The correct answer is are motor proteins that generate movements associated with microtubules. Both colchicine and colcimid block microtubule assembly of binding to free tubulin. In a motile cilium or flagellum, microtubules are arranged in this question we have already done. Now, why intermediate filaments? Intermediate filaments are dimers between 8 and 11 nanometer. Since this is intermediate to both actin as well as the recently studied tubulin, this is called as intermediate because the values are intermediary range, which is intermediate because the diameter of the two other principal elements of the cytoskeleton is there. Actin filaments about 7 nanometer and microtubules about 25 nanometer. So, between 7 and 25 you have 11, so it is called as the intermediate filaments that have diameter between 8 to 11 nanometer. In contrast to actin filaments and microtubules, the intermediate filaments are not directly involved in cell movements. To date, intermediate filaments have only been identified in animal cells. Intermediate filaments are strong, flexible, rope-like fibers that provide mechanical strength to cut to cells that are subjected to physical stress, including neurons, the muscle cells, and the epithelial cells that line the body cavities. Unlike microfilaments and microtubules, intermediate filaments are chemically heterogeneous group of structures that in humans are encoded by approximately 70 different genes. This I have taken from FABS Late Letters, a prestigious journal, and this journal gives a descriptive account of intermediate filaments. You can always give a general reading if you are interested to study further. Accordingly, intermediate filaments are everywhere, right from lens where you have facalin 6 and filcellin. You have epidermal keratins, keratin 1, 2, 5, 6, 9, 10, 14 and 17. You have simple epithelial keratin in the lungs, such as keratin 7-8 and 18-20. We also have on hair shaft and nail keratin 31 to 38 and 81 to 86. Nervous system has neurofilaments, the alpha internexin, nestin, perforin, GFAP, glial fibrillary acidic protein. The muscles contain cardiac, skeletal and smooth muscles contain desmin, syncolin and synamin. The blood vessel and other mesenchymal tissues contain vimentin, and finally the nucleus has laminin A and laminin B1 and B2. So these are the different types of intermediate filaments. Now we study what is the structure and assembly of intermediate filaments. You have an N-terminus and a C-terminus domain. It's a central rod domain containing of alpha helix with 310 to 350 amino acids. The intermediate filament proteins contain central alpha helical rod domains and N-terminal head and a C-terminal tail domains. Already I highlighted. The central rod domains of the two polypeptides wind around each other. See, coils or winds around each other in a coil structure to form dimers. There you have it, coils, and ultimately after coiling it forms dimers. Later the dimers will associate it in a staggered antiparallel fashion to form tetramers. Tetramers is 2 plus 2 tetramers, and tetramers associate end to end to form protofilaments, and laterally it joins to form filaments. That's tetramer joins to form protofilament, and protofilament twines around to form filament. Each filament contains approximately 8 protofilaments. Each filament contains 8 protofilaments wound around each other like a rope-like structure. It's like a coir or a rope-like structure that is formed. When you talk about the intracellular organization of keratin filaments, we have a GFP photograph, a micrograph of epithelial cells stained with fluorescent antibodies to keratin, which looks brilliant green. The keratin filaments extend from a ring surrounding the nucleus to the plasma membrane. Last but not the least, we shall study a bit about the attachment of intermediate filaments to desmosomes and hemidesmosomes. First we will go with desmosomes. This is the schematic representation of desmosomes, where the desmosomal cathedrins, that is desmoglein and desmocholine, I will highlight the desmoglein and the desmocholine. That's desmoglein and desmocholine. So the desmosal cathedrins such as desmoglein and desmocholine link the adjoining cells to the intermediate filaments. How it links? Through placoglobin, the placophilin and desmoplakin. Now we have the schematic representation of hemidesmosome. In hemidesmosome you have integrin alpha 6 beta 4, integrin alpha 6, integrin beta 4, and they will link the extracellular matrix to the intermediate filaments through plectin. BP180 and BP230, BP180 and BP230, BP230 and BP180 regulate hemidesmosome assembly and stability, and they are used for intracellular communication purpose. Now, what are the applications of intermediate filaments? Nestin is a stem cell marker. GFAP, glial fibrillary acidic protein, this can be used as an astrocyte marker in analysis of neuronal tissues. Vimentin, very important, is an antibody that can be used as a neural stem cell marker. Vimentin is highly expressed in fibroblast, and some expression is also found in T and B lymphocytes. The expression, they are expressed in many hormone independent mammary carcinoma cell lines. Vimentin maintains shape of the nucleus, and also it maintains the organelle positions, do not dangle and move from one end to another. So those were the applications of intermediary filaments. One last MCQ from this portion: which of the following is the most extensible filament? The four options are intermediate filaments, cytochrome, microfilaments and microtubules. Your answer obviously would be intermediate filaments. Finally, let me acknowledge the person, all those who have made this possible. Ms. Megha Dave, my student and current faculty, for preparing and compiling the CSIR net-based question banks. Dr. Darshi Bakshi, Associate Professor, School of Sciences, Navratna University, along with Dr. Elizabeth Robin, the co-coordinator, for hosting this program under the aegis of Gujarat State Biotechnology Mission. Ms. Ansuya Badalkar, Joint Director, HRD, GSBTM, for providing this platform, for her constant motivation and support in uplifting the biotech human resource across the state of Gujarat. And finally, with due acknowledgement to my guru, from whom I learned the tenets of life sciences, Professor A.V. Ramchandran, I would like to thank all the participants and august audience for your patience. Thank you very much.